ever think about, like, all the hidden tools that make big scientific breakthroughs happen? We're diving into one of those today. Antibiotics.
Okay.
You know, everyone knows antibiotics as infection fighters, right?
Right.
But get this, they're also essential for, for really cracking the code of life when it comes to molecular biology and genetic engineering.
Yeah, absolutely. You know, outside of just the pharmacy, these compounds can act as really precise selector in the lab. It's kind of like, imagine trying to find a single Lego brick, like a specific one, in a giant pile.
Yeah.
Antibiotics can help scientists do something very similar with cells and with DNA
Yeah.
when we make modifications to DNA.
Okay, so it's like they're gatekeepers when we're tweaking DNA in the microscopic world.
Exactly.
Today, we're going to be looking at how they're used in molecular biology, to help us understand how fundamental antibiotics are to making genetic research happen.
Totally. A key way this works is through what we call selectable markers during cloning and transformation. So let's say you want to insert a new gene into a cell.
Okay.
How do you actually figure out which cells successfully take up that gene?
This is where the antibiotic resistance genes come in, right?
Exactly.
Scientists, they add a gene that makes a cell resistant to a specific antibiotic, and they put it right next to the gene they're interested in.
Right.
And this is usually on a plasmid, right? Like
Right.
a small circular piece of DNA.
Yeah, a small circular piece of DNA. And so when you introduce these plasmids into a group of cells, like let's say E. coli bacteria,
Okay.
only the ones that manage to actually take up that entire plasmid, so that includes the resistance gene, will actually survive when you grow them on media that contains that antibiotic. And everything else, all the other cells that didn't take up the plasmid, they'll die.
So it's like a super efficient way to filter out the cells that didn't get the new DNA.
Exactly.
Only the ones that are genetically modified thrive. That's huge for things like basic gene cloning, making proteins, specific proteins, and even creating genetically modified organisms, right? This is like the secret sauce.
Totally. Think about it like developing recombinant insulin, for example. Antibiotic selection is a really fundamental step in identifying the bacteria that now contain the human insulin gene so we can make human insulin. Some really common resistance genes that you might encounter include AMPAR.
Okay.
And AMPAR confers resistance to ampicillin,
Right.
which is a relative of penicillin.
Oh, okay.
And it does this by actually producing an enzyme that breaks down the ampicillin.
Got it. So then there's CAN-R for kanamycin resistance.
Yes.
Kanamycin is an aminoglycoside antibiotic, and the CAN-R gene produces an enzyme that basically modifies and inactivates the kanamycin.
Exactly.
We also see TET-R for tetracycline resistance,
Yes.
and that one uses different mechanisms like pumping the drug out of the cell.
Right.
And then there's also CAT for chloramphenicol resistance, where an enzyme basically detoxifies the antibiotic.
Exactly. And what's really fascinating is that these same antibiotics, because they target very specific cellular processes,
Right.
they're also really valuable tools for studying how cells actually work. It's like using the enemy's weapons to understand their weaknesses, right?
Right.
Yeah.
So, for instance, rifampicin blocks bacterial RNA polymerase.
Right.
And that's essential for making RNA.
Exactly.
So scientists can use it to study the very beginning of gene transcription and the lifespan of mRNA molecules.
Right.
That's a really direct way to probe gene expression.
Totally. And then you have chloramphenicol and tetracycline, both of which interfere with protein synthesis,
Okay.
although they do it through slightly different mechanisms within the ribosome. But by adding those, researchers can quickly halt protein production
Right.
to investigate like protein turnover rates
Okay.
or the immediate consequences of a protein's absence.
Makes sense. And penicillin by
Right.
disrupting cell wall synthesis.
Yes.
So that rigid outer layer of bacteria, right?
Right.
That becomes a tool to study cell wall dynamics and even induce bacterial cell lysis.
Exactly.
Like each antibiotic is a key to unlock a specific area of cellular function for study.
Absolutely. And it's worth mentioning, although less commonly used, sometimes specific antibiotic or chemical treatments can even be used to actually eliminate plasmids from bacteria.
Oh, wow.
It's a process called plasmid curing.
Okay.
So you can get rid of the plasmid if you want.
So the big picture here is that antibiotics are far more than just infection fighters.
Absolutely.
They're really indispensable behind-the-scenes enablers of molecular biology and biotechnology.
Right.
They're not just treatment.
Yeah.
They're foundational tools of discovery.
Exactly. They act as really essential selective markers,
Right.
which allow us to actually pinpoint and study genetically modified cells. And they're also really powerful research probes to help us understand those intricate mechanisms of life at the cellular level.
It really makes you think, like, how this seemingly simple application of antibiotics has paved the way for so many scientific advancements in medicine and beyond.
Yeah.
What future breakthroughs, we wonder, will hinge on these very same fundamental techniques.
Right. Who knows?
Thanks for joining us for this deep dive.
Thanks for having me.
We'll see you next time.